Yes, welcome to the day 8 session on QSAR, Pharmacophore modeling as well as Pharmacokinetic modeling. So we'll be discussing about these topics on today. Before going to today's session, as usual, can we go to the last session topics? We can recap the last session topics. Yes, we discussed on force fields, what is force field and what are the types of force field, and then we have discussed on molecular mechanics, then we discussed on quantum mechanics. Okay, what else did we discuss other than this? Probis. Yes, you are right. We have discussed one server called Probis server, Probis charm server, where here, whatever we work with Probis, I show you the output now. The result got generated on yesterday. So you'll be getting like this information, like Probis finished. Results are available in this. And there is also one more Probis results. So this is the hyperlink. Because I have mentioned email ID and whatever results we generate from Probis, okay, we can able to have in repository for not more than a week. So if you want to save the results, you better save these results, okay, in the form of snapshots or in the form of PDF documents. So if you see here, I have given an input of 4W05. This is the PDB ID of a protein. So for the protein, whatever I have uploaded, okay, we can able to get, okay, and also I specifically mentioned the A chain. For this, we got the structures of compounds, okay, and the corresponding protein PDB IDs for whatever protein structure we have searched, related kind of a protein structures we can able to see, and what are the compounds best fit with the protein structure. So how we can able to identify whatever we identify, okay, this is represented in terms of confidence scores. So this is something different, confidence. Generally, if you see in docking, we measure the binding of ligand with a receptor through binding energy, isn't it? Binding energy in terms of kilocalorie per mole. But the affinity of the ligand with a receptor, here the confidence is something related to pharmacophore. In the pharmacophore modeling, where we get these kind of confidence or fitness scores, there is a slight difference between the pharmacophore modeling and docking. In pharmacophore modeling, we just check the fitting, whether how effectively this ligand is going to fit. But whereas if you see the docking, we check through different conformations and we use the chemical chemical conformation similarity, etc. So tomorrow anyhow I'll be discussing on molecular docking. So we discuss extensively on molecular docking on tomorrow. Okay, a slight difference is there in the pharmacophore modeling as well as molecular docking. One thing you have to remember, in pharmacophore, we get fitness values or confidence values, but whereas in the docking, we can get in the form of binding energy. So binding energy, that is functional group of ligand, functional group of receptor, that is most important to determine the value of binding energy. But whereas here you won't get. Here you just check like whether if pen cap is closing the pen or not. Just like that fitting. The fitting alone you will be checking here. Got it? Now we understand. So more if you say, discuss on tomorrow session, you can understand more on talking now. So binding sites, there are multiple binding sites. Okay. So for each and every binding sites of the proteins, we can able to get the corresponding kind of ligands. What ligands are no dock with what kind of similar proteins. Okay, so this is for Probis charm. Okay, so then we have discussed something on types of MD simulation and applications of MD simulation. Okay, so how the MD simulation can be applied. In that, we have also discussed on Monte Carlo simulation. Then we have gone across a type of drug design called de novo drug design. In the de novo drug design, we have discussed two types of de novo drug design. One is ligand based, the other is structure based. In the ligand based, you have further have classifications. If you see in this de novo drug design, especially, you can find that fragment based drug design. Okay, that is the most important. Like we have steps in de novo drug design. Okay, so and we can also see how multiple fragments of the drug is connected as a single drug. So all these things we have discussed in these two types of de novo drug design: de novo linking and de novo evolution. You remember these are the two process we have taken in the we have discussed in de novo drug design: de novo linking and de novo evolution. And then we have discussed something on siderophores. Siderophores or iron chelator molecules. The drugs are conjugated with the iron molecules. Especially, I told you if you want to treat Pseudomonas aeruginosa infection in lungs. Okay, so targeting the drug through siderophores is an effective strategy for reducing the infection. Okay, so the that we have discussed and we have linked with the de novo drug design. How to design the siderophores? Of course, one should have a knowledge on de novo drug design to design a drug for this antimicrobial activity. And then, if you see this, what is the range of good confidence score? Oh, you're asking the question range of good confidence score. The range actually it comes around 5, I believe. Okay, maximum might be the 5 confidence score. But I did say I see up to the 5. I see less than 4. So the confidence can be 4 also. Yeah. So I didn't see 5. So the maximum confidence range will be in the 5 up. I mean up to the 4. Up to the 4. Okay. Then anything else you want to discuss? So till now, whatever we have discussed, till now, whatever we discussed on yesterday, we have completed. Now, let me take you to the today's session. On today's session, we can able to discuss on 3D QSAR, QSPR, QSTR, Pharmacophore modeling. Now, let me take you to the QSAR. What is QSAR? QSAR stands for Quantitative Structure Activity Relationship. This is purely a ligand based drug design. This is actually ligand based drug design where the 3D structure of the ligand is sufficient enough to work on determining different properties of the drug. Okay, here in this case, QSAR, the major property what we discuss is the activity. So in QSAR is a derivative of QSPR. Okay, quantitative structure property relationship. If property is activity, then we call that as QSAR. So like this, we have different types of properties for the compounds, for the ligands, for the drugs, for the agonist. So this is what we discuss in this session. And we also discuss on statistical parameters in QSAR modeling and types of molecular descriptors and the softwares used in QSAR, pharmacophore modeling. Then we also discuss the pharmacokinetic modeling, which is of course essential for drug design. Now let me take you to the QSAR. QSAR, that is quantitative structure activity relationship models, or a regression mostly regression, no, regression or classification models used in the chemical and biological sciences and engineering. So here if you see in the QSAR regression model, we have predictor variable X and we also have a response variable Y. Okay, so while classification to QSAR models. And we generally relate chemical structures and biological activity in QSAR models. So more about this we see in the subsequent slides. History and development of QSAR. So if you see QSAR began from the history began in 1868 by Brown and Fraser. They brought this kind of a function, a QSAR function, that is the assumption of physiological activity phi is a function of the chemical structure C. If this is from Brown and Fraser, Mayer and Wharton got the lipoid theory of necrosis. But the exact kind of a birth of QSAR came from Hammett. He is the most important scientist. So whatever parameters we study in QSAR related to electronic sigma constants, okay, this was discovered by Hammett. So the origin started in 1930, but the term was proposed by Hans. Okay, that is a QSAR term was proposed by Hans in 1964. You know the birth of computer, when the computer is born in 1950s, and birth of Unix. So this also in 1959, 1960s. So birth of C language, I mean before the D language, everything in this 1950s, 60s like that. So at the time itself they got, so whatever the QAs here, they got that. But whatever we are implementing in computational, it happened in 1988. So if you have a 3D structure of a QS, I mean a 3D structure of ligand, then we can work on 3D QS here. Okay, so meanwhile you can see these developments. Andrews brought the affinity contributions. There are different types of QS here. Okay, we have from 0 to 60 of QS here. So we have some types of QSR based on dimensions. Okay, so we'll be discussing in subsequent slides. So this is something about the history and development of QSR. Recently, we also discussed on Lipinski. You remember? So this was brought by the scientist Lipinski in 1997. Majorly, this is used to check the bioavailability. That is based on four rules. That is based on four properties, physical and chemical properties of drug. We can able to check whether the drug is best for oral bioavailability or not. If the drug has to be taken in the oral route, whether it's having a good oral bioavailability or not. So this can be determined from the Lipinski rule of 5. Okay, and similarly can see a drug-like character in 1998. About QACR, it is a ligand-based drug design, and this was proposed by Hans and Fujita, as I mentioned you earlier. It possibly aims in establishing the possibility of a relationship between a set of descriptors. Majorly, we study the electronic descriptors, and we link these electronic descriptors through pharmacological response. Okay, so what are the general parameters of the chemical? So if you take a chemical API drug, even just 5 minutes before, HR from a pharmaceutical company contacted me. Sir, I have multiple vacancies. If you know any of your contacts, kindly tell me. Even we are ready to come to your college for placements. See, there are plenty of startup, pharma startups are emerging or originating. So plenty of opportunities are there. They need this kind of knowledge also. Okay, so like just drug design is not sufficient. One should also have something about drug development. If you try to develop a drug, what are the parameters? What are the physical chemical properties we analyze? So whatever we work in computational, how we can able to work with an in vitro. So these kind of a correlation kind of a studies also help you. Okay, QSR is purely a computational approach. But however you want to calculate the activity, to calculate the activity, of course, we have to use the wet lab experiments, isn't it? And similarly, if you go to the protein-protein interactions, we have seen StringDB and other databases. How this is established? Only through immunotechnology experiments. Again, the wet lab experiments data is what we are using in computational data. And we're just checking whether the in silico and in vitro data are matching with each other or not. Finally, we correlate these kind of data. Okay. So now let me take you to this IC50, EC50, Ki value, inhibitory constant value. So these are important to calculate the biological activity. What biological activity can say types of biological activity. If you're working on antimicrobial activity, that is, if you want to reduce the Pseudomonas aeruginosa infection through siderophores or any series of drugs, whatever we design. Okay, then it is an antimicrobial activity, isn't it? How the antimicrobial activity can be determined through the zone of inhibition. So antimicrobial activity, whatever drug you have, we can able to prepare it in disk, and if you place the disk in the specific pathogen or specific bacteria where you want to check the biological activity, antimicrobial activity, we can able to see from the zone of inhibition. Generally, the zone of inhibition, whatever I mentioned, okay, so this is actually used to calculate the activity. You know how this is calculated? The negative logarithm of zone of inhibition, generally in some cases they used to determine activity. We have different correlation formulas to calculate the activity. Under an antimicrobial study, we can use these kind of a formula to calculate the activity of the, that is antimicrobial activity of the compounds or drugs, whatever we have developed. Similarly, we can also check the activity through LD50. LD50, remember, lethal dose 50, AD50. Generally, we use it for animals. If you want to test the activity of drug in small animals, generally we use this LD50, AD50. So whatever we use these kind of parameters, this can also be used, similar kind of a correlation can be used to determine the activity. So we have different kind of formulas linked with the physicochemical properties, and even through the physicochemical properties, one can able to check whether your compound is a toxic or non-toxic, active or inactive. This can also be determined, and also whether it is a mutagenic or non-mutagenic, whether it's a carcinogenic or not carcinogenic. So all these biological activities, okay, categorical kind of an activities can be determined using this QSAR. If you go to the Protox 3, in Protox 3, we are not just getting the LD50 dose alone, we are also getting whether your drug, whatever we input, is a mutagenic or not, whether it's a carcinogenic or not. So like this, we can also see even the toxicity with respect to specific organs, isn't it? So, whatever we see in the tools, databases, okay, so of course, we can use this kind of a data to use this ligand-based drug design QSAR. Then, how to use this data to in QSAR to determine the activity? That we'll be discussing now. So, QSAR, we have basically two important terminology. If you see in docking we have terminology like ligand and receptor. Ligand is generally a drug and receptor is generally a target. Similarly, if you see in QSAR, we have two important terms. The most important term in QSAR is number one is training data set, and the number two is test data set. So in training data set, okay, if you know the activity of 500 compounds or some more than 10 compounds, okay, if you go with more than 10 compounds, and in my case if I have 500 compounds, okay, that is activity is known for all the 500 compounds, but if you don't know the activity of the test compound, the test compound is something recently developed, I don't know the activity. If I want to check the activity of this test data set, this we can able to use from the training data set through interpolation or extrapolation principles, that is through linear regression model. What is linear regression? We'll be seeing in the subsequent slides. Either we can use linear regression or classification. I told you earlier. So one the activity of unknown test compound can be determined from the already predicted activity of 500 compounds. Okay, so this kind of a concept is generally a kind of a machine learning approach, isn't it? It's a kind of a, we use a different kind of a machine learning approaches for estimation of the activity. So if you see these validation parameters in QSAR modeling, one can use the method of least squares, cross validation, adjusted R2, chi-square test, root mean square error, bootstrapping, and scrambling. So these are different validation parameters in molecular modeling. Types of molecular descriptors. So if you see the molecular descriptors, we have experimental measurements and we have theoretical molecular descriptors. Experimental, we can able to determine log P, molar refractivity, dipole moment, polarizability, and in general additive physicochemical properties. So all these measurements we can able to measure in our lab itself. That is for a drug, if you have a drugs, we can measure all these parameters. But there are also theoretical molecular descriptors. Okay, what are that? Symbolic representation of the molecule and can be further classified according to different types of molecular representations. Okay, so the symbols we use, okay, is what is a theoretical molecular descriptors. And if you see these classes of molecular descriptors, to know more about this, you can see this classification. 0D descriptors. This is constitutional descriptors, count descriptors. Structural fragments of the drugs, also called as fingerprints. Okay, this comes under 1D descriptors. If you have a graph invariance, 2D descriptors, and 3D Morse descriptors, Wim descriptors, they're like this size, steric, surface, and volume descriptors are available. 4D is something related to grid, and this can be found out from COMFA or COMSIA methods. So these are the methods of QSAR. Okay, these are the tools we can use. COMFA, COMSIA. These are the tools used to work on QSAR analysis, including volume surf. Okay, so this is something about 4D descriptors. So much of theoretical information is there, isn't it? ChemDes is one of the molecular descriptor and fingerprint computation. So just in Google, if you go with the ChemDes, you can find the types of descriptors you see here. This is what built in the software. There is something called chemopy descriptors. We have around 1135. You know, what did the scientists know what exactly is the difference between all these 1135? CDK descriptors 275. Okay, so the scientists have developed different kind of a descriptors. A number of descriptors are shown here. Molecular fingerprints. That is, you know, each and every molecule can be distinguished with the fingerprints. Like if you take withanolides, I told you in withanolides we have 41 withanolides, around 41 withanolides in ashwagandha. You know, most of the withanolides have similar molecular formula or similar molecular weight, but these are distinguished through the fingerprints. Even the canonical smiles format can able to distinguish all these 41 compounds. Okay, so like this, we can able to see information of the descriptors from 0 to D to 3D and also up to 4D. We say first initial how we can take steps. First, we should know the molecular structure. The molecular structure, you can have any of these dimensions. You can have even if you have a 0D, that is molecular structure, we can still work with QSAR. But it is better to go with a 3D or 4D. That gives more precise information. Okay, so just these coordinates, 0 to 40 coordinates of the molecule can be used to determine and we used to determine the molecular descriptors. I mean, actually, for the compound, we can select what are the important molecular descriptors required. For example, if you are focusing on brain targeting, or mitochondria targeting, lipophilicity is one of the most important parameter. So based on the application, what exactly we are targeting, okay, so based on that, we give the importance to the molecular descriptors. So we give the importance based on the priority, which is most important. We give that information priority to that. So there is no need to select this much descriptor. See, I told you we have 1135, 275. No scientist can use all the descriptors, but what descriptor you know already, or whatever present in the demo file, you can use that as a default, and you can able to determine the activity, of course. Multivariate analysis, whatever molecular descriptors we use, we take it for the multivariate analysis. So, like if you have a multiple compounds, and if you are focusing on multiple properties of the compounds, so you are taking multiple compounds, multiple properties, then obviously we go with multivariate. So multiple linear regression, principal component analysis, machine learning techniques like neural network, support vector machine, k nearest neighbor, self-organized, all these are a machine learning techniques, isn't it? We can use the machine learning techniques to determine even the activity of inactives, to determine the activity of inactives. Generally, we use the machine learning technique for determination of activity of inactive compounds. And if you see this correlation to activity, okay, we can have R square, Q square, R square. So these are some of the parameters we can able to use it to correlate the activity of the compounds. Generally, if you see the compounds, if I have nearly some 12 compounds or up to the 15 compounds, whatever compounds I have, this is generally plotted the response versus the response in terms of experimental activity and a variable that is predicted activity. So y versus x, you can find the compounds based on the activity data. You can able to find these dots. And obviously, if you go with a regression, we check in which dots you can find a line. If you see this, this is also giving a straight line, and you can also check what can gives the 3 points are joined here. And if any 3 points can able to join, you can check which is the best line. So whatever line I have drawn, okay, this kind of a straight line, this is actually y equals mx plus c. So that is the response y, okay, is in y-axis, and of course the experimental activity is y, then predicted activity is x, and so whatever slope you get is m, and the intercept where is going to intercept, it is going to intercept is c. So this is how we can use the regression model to check the line of best fit. So through the best fit, you can able to check what is the accuracy of prediction of compounds. If you try with minimal compounds like 10 to 15 compounds, whatever activity you predict for test data set is not accurate. Okay, so the even the R square value, you can see deviation. You can see it's not close to one. It won't come close to. If the R square value is close to one, then you can tell you have a good kind of model developed. Okay, to check the model, to build a good model, we should have more compounds in the training data set. If you have more compounds, can be 500 when compared to 500 compounds. In a data set, if you have 1500 compounds, that 1500 compounds can give a better kind of R square value, a better kind of an accuracy of prediction values. So like this, we use this kind of a machine learning technique for the prediction of activity of unknown compound, or also called as test data set. Okay, and finally we can validate the model through residual analysis, external validation, receiver operating characteristics, ROC, Y randomization testing. These are the softwares, types of QSAR softwares, a number of descriptors used. So if you are focusing on to use many number of descriptors, better go with this E-dragon. QSAR related terms. Like QSAR, I told you, A stands for activity, but the general term, how it is derived only from QSPR, where QSPR stands for quantitative structure property relationships. And if the property is retention time, you remember the retention, where you find this retention? Can someone tell me what do you mean by, what do you mean by QSRR? Here they have mentioned like retention. So where you can find the retention? I also mentioned you something like retention time, a clue I've provided. Where can we use
Chromatograph.
QSRR?
these chromatographic techniques.
Exactly. Exactly. Your answer is right. If you take ethanoids, I told you we have 41 types of ethanoids. So, all these 41 ethanoids have almost a similar kind of a molecular formula or weight. Then how we can determine? How we can able to determine? So, even if you can able to separate, distinguish compounds through retention, okay, if you want to separate more closely related compounds with respect to formula, size, etc., then obviously we can use chromatography. This is more defined when compared to retention. In retention, we have different molecular weights. Compounds can be separated. But if you have similar molecular weight, molecular size, obviously one has to go with this quantitative structure chromatography relationships. So both are used in, of course, in chromatography. And quantitative structure toxicity relationship, wherever you find toxicity, how the toxicity is determined? The toxicity is also determined only through the physicochemical properties. In Protox 3, you are feeding the smiles format, but you are getting the toxicity with respect to all the organs. How is it predicted? What is through a kind of a correlation of a physicochemical properties? Okay, formula is used, and through the formula they have estimated in competition. So, even the toxicity, okay, so however we calculate the toxicity of different compounds through computational, if you have developed already a training data set, even test data set can be predicted through that. Okay, so then similarly, for toxicity, we use the partition coefficient, molecular weight. This is general property, isn't it? Even this general property is used to determine the toxicity, whether the compound is toxic or not toxic. Okay, so one should know about the chemistry. Based on that formula, one can also find the toxicity. Even plenty of jobs are there who are working on toxicity, can be in silico, in vitro, and in vivo. Now, even recent recruitments are happening on to develop the software for toxicity. Okay, in Gujarat. Okay, now let me take you to this quantitative structure electrochemistry relationships. Electrochemistry, like if you're focusing on developing corrosion materials, okay, I mean the anti-corrosion materials to avoid corrosion, especially those are working in marine. There are different kind of equipments, different kind of thing they use in marine, isn't it? So if they want to, the electrochemistry is most important in that case, just because they find more salt in the water. So obviously, if you work on the anti-corrosive kind of materials, one can use QSAR. And similarly, quantitative structure biodegradability. So we have plenty of information. I mean, plenty of applications are there in this ligand-based drug design. QSAR, generally one thing you have to remember in the QSAR is the activity is generally predicted through function of physicochemical properties and or structural properties plus error. A bias can be included. I told you even toxicity is determined like this. Toxicity is one of the activity of compound, isn't it? So this is the general formula. And 3D QSAR. What is 3D QSAR? How it distinguishes with the normal QSAR? 3D, the acronym is clearly representing we should need the 3D structure of the chemical compounds of the small molecules, whatever we use. So we use the 3D structures, of course. With the 2D structures, you can't able to work. You must have a 3D structure of the ligands. And here we can use the tools like COMFA, that is comparative molecular field analysis, and also COMSIA for determining the activity of a test data set from the training data set. So this is a workflow for predictive QSAR model. Okay, so descriptor generation, split into training data set and external validation sets. Use the appropriate machine learning techniques, neural networks, support vector machines. Use any of the appropriate or a combination of machine learning techniques. Select the models with high internal and external accuracy through R square value, ROC, and assessment of applicability domain. So later you will be assessing whether based on the accuracy prediction, this model is good or not. So overall study design. So almost we can use in different applications a similar kind of a methodology. QSAR protocol. So each and everything is taken from different journal papers. Okay, this is a machine learning tool for QSAR modeling. What are the different machine learning tools available for QSAR? So R, Rapid Miner. Myself, I tried with Rapid Miner in 2014. Okay, so the most of the tools are commercial softwares. Okay, Rapid Miner 9. R, we can use it for free. Okay, R we can use, Vika we can use. So in these tools, we can have the QSAR. Galaxy, this is an online tool. So to work with the sample files, to initially work with the sample files, one can choose this kind of server also. Later, you can build the tools. Okay, in a cloud platform, you can build the training data set and then you can check the test data set values. So the steps, whatever we discussed, build the QSR models. Here they use a model called MQSR, MCQSR, how to build and how to predict. And so these hyperlinks are mentioned to work on QSAR. So this is how a linear regression model looks like. If you have a data set of around 15 compounds and you try to see fit a line, this is a linear regression line. So you try to draw a line where it can able to join maximum number of compounds. This is the line of best fit actually. For this best fit, we got R-square value as 0.906. So if Oscar value is close to 1, if it is, if you tried with some 100 molecules instead of 15, and if your Oscar value is increased to 0.92, then obviously you can able to tell this data set, whatever the accuracy you predicted is good when compared to this. You can follow with the 100 compounds instead of 15 compounds. So one should remember that increasing the population of training data set can increase the R square value. And this kind of a model, we call it as a linear regression model. Okay. So this is something about an output from the galaxy ASCR. Now let me take you to the next concept called pharmacophore modeling. Till now, we discussed on QSAR, types of QSAR, and tools used in QSAR, and how an output of QSAR a training data set looks like, and how the test data set can be predicted. See, if I have a compound somewhere here, if my one compound, the test data set is somewhere here, okay, then obviously, okay, whatever experiment value we have predicted, okay, from that, the log IC predicted value can be determined. Okay, so if you know this, so based on this interpolation or extrapolation, okay, we can able to get the kind of value. Okay, so if your data is lying here, then obviously you can check the value is zero point 0.9. So like this, if we should need first of all y equals mx plus c line in linear regression to determine the activity of unknown compound or test data set. Okay. Now let me take you to the next concept, pharmacophore modeling. What is pharmacophore modeling? A pharmacophore is an abstract description of molecular features that are necessary for molecular recognition of a ligand by a biological macromolecule. Actually, in the steps in drug design, initially we start with hit. From hit, we try to develop a lead. This is the backbone of a drug. From the lead, you develop so many lead candidates. And if you want to screen the lead candidates, we can go with the pharmacophore. Or after the lead, we can also use the QSAR. But after the QSAR, one has to go with this pharmacophore modeling. And after the molecular for the pharmacophore modeling, one can go with talking. And after talking, one can go with the simulation. So this is how we can use this computer drug design in sequence. Start with hit, and then we use the different methods to determine to develop it or design a drug. To design a drug, we use this kind of a lead. Here I mean lead generation. Pharmacophore, here I mean like pharmacophore modeling. If you need mapping, we can go with pharmacophore mapping with multiple compounds. Molecular docking can be done one ligand with one receptor. But if you have multiple ligands and if you want to screen it, we can use virtual screening based docking. Virtual screening based docking can dock multiple ligands with the receptor. Then finally, whatever the best docked ligands, we can go with the MD simulation. Okay, so we start with the hit and then we complete up to the stability. We can check the stability of the ligand or a drug through this sequence of drug design methods. Okay, so if you see this pharmacophore and if you see the docking, I told you in pharmacophore we determine this molecular recognition through fitness values. Fitness value based on the fitness value, like however, I think a participant Vishnu has asked what is the threshold, sir? Yes, of course, each and every tool have a threshold value. You don't have more than that threshold value. I believe it is 4. Even for whatever tool I'm going to show, the next tool what I'm going to show is the form mapper. For that also use this kind of a maximum fitness value as 4. So we can check how close it value you'll be getting when a drug, whatever an input you feed in this pharmacophore to get, which is the, that is, if you feed a drug, you'll be getting plenty of macromolecules. The drug is fit with best fit with what macromolecule that will be ranked based on the fitness scores. Got it? So this is something about pharmacophore modeling. Now let me take you to the pharmacophore modeling. In pharmacophore modeling, if you have a backbone, so the you have we have many functional groups. If you see in polar compounds, the most important functional group you find in polar compounds is OH. So you find OH. So this OH can link with any kind of polar amino acids of a protein. Polar group have a binding affinity with the polar group of amino acids. If this is your ligand, polar group, you understand polar always dissolves the polar. Non-polar always link with the non-polar, isn't it? So this is how the interactions also will be there. Okay, it can be a docking or pharmacophore modeling. So one should first find the activity of the functional groups in the lead compound. If this is your lead, so if this is your lead, okay, we can attach some functional groups and we can check. So for the functional groups, we can able to see if the fitness values increases based on the attachment of functional group, then we check what functional groups are important to design the drugs. Okay, that functional groups, of course, that is a new molecule you have constructed and we can use it further for uh, drug design. Okay, now let me take you to the typical pharmacophore features include hydrophobic centroids, aromatic rings, hydrogen bond acceptors or donors, cations and anions. Okay, so generally in this pharmacophore, I told you we come across with lipophilic or hydrophilic. These are the most important interactions we check in pharmacophore, and based on this, the functional groups are attached. Whether you can attach CH3 group, the CH3 group is non-polar. If you attach OH, it is polar. If you attach NH2, it is non-polar. So what functional groups we are attaching? If you are attaching OCOCH3, how about that? If you attach COOH, how about the lipophilic and hydrophilic? Like this, we try with different functional groups in this pharmacophore modeling. Use a lead backbone compound, attach the functional groups, and check the fitness values. So if you see these steps in model development, similar to QSAR, we select a training set of ligands. Then we go with the conformational analysis. Then we go with molecular superimposition. So basically, it defines with QSAR, like here, other than checking the activities, we are also binding with the receptors, binding with the targets, and then we check the fitness values. Okay, so next to the QSAR, we go with this method. That's what I'm telling. Okay, according to the sequence in drug design. So steps in model development, abstraction, validation. This is important. And now let me take you to a tool called Pharmapper. How to work with a Pharmapper. So in Google, type Pharmapper. So if you will be getting this kind of a home page. In this home page, you have to upload any drug compound. So actually, I have uploaded a compound called boyrevinone. Boyrevinone B. Boyrevinone B is a rotinoid compound from Boerevia diffusa. Rotinoid, it means isoflavone. So this compound, I just worked on the treatment of different diseases. So whatever Boyrevinone B compound, if I try to upload it, okay, we'll be getting this fit scores. See, the fit score is 3. Then it's not a 4. The threshold value here is a 3. The threshold value, what we got is 3. So the maximum thing, whatever we got like 1. Okay, if a normalized fit score is 1, the threshold value is 3. You got it? So it depends on the tools what we use. And you don't find any target name, but this is the PDB ID. If you search PDB ID, you can get the name of the protein. But this pyrimidine B, it's based by binding with the second thing, DNA directed RNA polymerase. These kind of polymerase enzymes are present in viruses. So this can use as an antiviral compound. That's what we got. And simple as we can also check the other kind of receptors. Okay. So what is it based used? What is it? What are the biological activities of the compound? So this can be determined only through fitting a compound with the target receptors. This is what we call it as molecular recognition. Through molecular recognition, one can able to determine the type of protein molecules and even the bioactivity can be determined accordingly. Target deconvolution kind of an approach, isn't it? Form mapper. So form mapper, I told you, according to this tool, we are through using the threshold value, we can able to find, okay, what is the most appropriate binding receptors. If you see here for each and every receptors, you can see your ligand pharmacophore. So this is the backbone. The blue can be a backbone, and whatever gray color shown are the functional groups attached. Attaching and detaching can also variate the kind of thing. But already we have fixed the ligand molecule as BB. Okay, if you have any other derivative of borevinone, instead of borevinone B, if you give borevinone A, you can check what is, how about the, based on the fitness values, you can check whether it is going to have antimicrobial activity or anti-cancer activity. So just the functional group, 1 or 2 functional groups in position can change the biological activity also. Got it? So this is something about pharmacophore mapping, and this can we can do with pharmapper. Pharmacokinetics, there is a lot to study. Of course, pharmacokinetics is also an important for drug design. Pharmacokinetics is the study of how an organism affects the drug. If you want to work on pharmacokinetics, myself, I am working on MATLAB. So MATLAB is the best tool to work with pharmacokinetics. Okay, so pharmacokinetics, pharmacodynamics. We already discussed in the previous sessions that if you focus on the mechanism of drug with respect to treatment of disease, pharmacodynamics, both pharmacokinetics and pharmacodynamics, a part of pharmacodynamics, we can able to work with MATLAB. Okay, but in pharmacokinetics, we use pharmacokinetic models. What are the types of models available? So types of pharmacokinetic models, compartment models, it depends on the type of work. Okay, this is a major classification: compartment, non-compartment. In compartment, we have physiological model, mechanistic model, and in physiological further classified into three types of models. So metabolism model comes under physiological modeling of compartment. So what is then compartment? What is non-compartment? We'll be discussing more about that. So if you see this kind of diagram in this figure, okay, so the blood actually flows from arterial to the venous in different organs. If you focus on liver disease, how about the dosage pattern? Whatever dose we give in oral route, how much bioavailability can find in oral? But if we want more kind of a bioavailability, instead of giving a oral, we can use IV dose. Okay, directly it takes in the vein. So one should remember about that. And if you see these compartment models and non-compartment models, in compartment models can be classified based on the type of number of compartments. Here, first we are discussing on one compartment model. In one compartment model, if you're focusing on uh, uh, if you are focusing on the blood as the main compartment. For example, treatment of common fever. In treatment of common fever, okay, that is to relieve from phyrogens, we use a drug like acetaminophen. The acetaminophen is going to target, of course, COX-2 enzyme. The COX-2 enzyme is present in blood tissue. Then compartment 1 is the major tissue or organ we focus. So whatever acetaminophen we take, how about the absorption and how about the elimination? Generally, the adults of 10 to 14 years, they have adolescents in the age of 14, 10 to 14, they have very good metabolism. The absorption is rapid to them because of hyperactivity we can observe from them. And of course, this kind of availabilities and even the half-life estimation of the drugs, okay, we can able to determine through different properties. Okay, three compartment here instead of a single compartment like a blood. If you are focusing on, if you are focusing on, for example, obesity. For obesity, one can able to determine through fat accumulated like adipose tissue and even based on the muscle. Okay, if you have more muscle, we obviously can tell the obesity is less. Now, obviously, if adipose tissue is also less, we can tell they are very fit, isn't it? So, if you are focusing on kind of obesity or linked with diabetes, obviously one can go with these three important compartments. That is compartment, central compartment. This is compartment one, and this is compartment two, and this is compartment three. Okay, so like this, like one can able to work with multiple compartments if there is a necessity to work on like whatever drug we provide, whether it's targeting all these organs or not. So there is a plenty to learn about these types of. If there are no compartments, we call it as non-compartment model. Then how to work in computational part? That's most important. If you want to work these pharmacokinetic models, we can work with MATLAB tool SimBiology. So SimBiology is the tool present in MATLAB. From this, we can able to work, and these are the 5 steps in SimBiology. Fortunately, Sri Venkateswara College of Engineering have MATLAB license. Not only the faculty members have an individual MATLAB license, even each and every student has a MATLAB license. So if one want to work with the pharmacokinetics, okay, we can use this MATLAB. Of course, in MATLAB we have a unique kind of a programming, MATLAB programming. But for in this case, there is no need to use much of the programming code. We have already developed data sets, I mean demos. Using the demos also, we can able to use it for our applications. Okay, and if you see in the steps in SimBiology, building model, simulating model, estimating parameters, analyzing model, deploying model. This is similar to the machine learning technique, how we make it, isn't it? So building model. For building the model, that is can be a pharmacophore based pharmacokinetic models, or pharmacokinetic or pharmacodynamic model, okay, we can use system biology markup language. Okay, the system biology is what we are using to determine the pharmacokinetics of a drug, okay, including pharmacodynamics. So we can create the models using the variants, and then we evaluate the dosing strategies. For example, if you come across paracetamol, 3 doses has to be taken that day. That is, out of 24 hours, you can see 3 dosage patterns. So you can find up to this for a day. Sorry, the line is not straight. You can take an erase. See, if you see this kind of a line, okay. So up to this is 24 hours, and this might be is prolonging to the 48-hour period. So how many doses? So initially, you see after either this might be in the 6th hour or 7th hour, the dosage is gradually reduced. And then if you take the second dose, again the viability is increased. And then it's going to decrease within 6 hours, 6 to 7 hours. Then again you take another dose. So like this, we can evaluate the dosing strategies. Steps in symbiology tumor growth fit model. So how the tool looks like. We have a central compartment, peripheral compartment. Okay, so we can draw number of compartments, and we can each and every compartments we can able to fix the rate constant. How much stock is going to pass it to peripheral, and how much is going to eliminate. So all these things we will be feeding this. This is like an artificial intelligence. We have to feed all the parameters. We have to fix the design, fix the model, and we have to even have a range of values with respect to even rate constants, concentration of drug, etc. Okay, so all these things you can also feed it here. So you can able to feed it here. Okay, these are the properties you can see here. And after if, there is already this is already a demo file, simply we have to load it. And if you click model analyzer, it gives you this kind of a window. So what drug and how many doses and what type of dose, these parameters are already fixed here. You have to simply click run. If you click run, you'll be getting this kind of a table kind of a data. This table data, we can see the R square value. So R square value we got is 0.9934, more accurate. If it is nearing to the 0.99, this is the best kind of a model. Okay, so this we have already found from the demo file. So if you click the analyze button, if you click the run button, finally we'll be getting this kind of a curve. Whatever the output I am showing is a tumor growth model. Is a tumor growth model that is for breast cancer. For breast cancer, if a patient is taken paclitaxel of a particular dose, even the dosages are mentioned there. So if paclitaxel is taken in a particular dose, whether it's going to treat the breast cancer or not. So this can be checked over a period of time. So this is checked from 0 day to 40th day. 0 day to 40th day, and the blue color line shows the central drug and the red color line shows the peripheral drug. Okay, so this is something about how to use. Now let me take you to, just I need 1 to 2 minutes to take you to the form mapper. So we already seen the probes, isn't it? 1 minute. I have already worked on form mapper. I didn't get the result in form mapper. So for the form mapper, you have to simply go to the Google, type form mapper, okay, and then this is the URL form mapper ECST. Uh, Inside that, you have to submit the job. Okay, okay. The continuation of usage of MATLAB will be discussing on tomorrow. Okay, like even if you have a time, we can discuss. I think I can join. So just because you're in, I need just 2 minutes because I found many participants are going out. So just provide me 1 or 2 minutes to explain how to use the MATLAB. For FileMapper, simply go to submit job. Inside the submit job, upload the ligand file in STF format. So to upload the ligand in STF format, so we can able to download the file STF format from PubChem. So I just uploaded that ethafrin a.stf. So provide the email ID because it takes some time to generate, and then give a job description and then click continue. So immediately you won't get the result. This is getting submitted, but it's asking you what kind of a target data sets. Choose human protein targets, all targets, whatever you want. If I want druggable pharmacophore model, in default, I keep it as such. And in a day, you can't submit more than 10 jobs. So already I submitted one job. Now I am submitting this as a second job. So you won't get the result, but after some time, if you get a result, the result link you can able to find in your email ID, whatever we have typed. Now let me take you to the MATLAB. There is much to learn here about the pharmacokinetics. So whatever I explained in the PPT, okay, just like that you have to try with the MATLAB. So now let me take you to the MATLAB. The recent version I installed is MATLAB R 2024A. Okay, in the MATLAB, even for the newcomers, you can able to learn the MATLAB from the help manual, etc. Also, we have some courses like Coursera, Udemy. Here also we have MATLAB courses. If you have a license, even you can able to complete these courses free of charge. The e-certification will be provided after completion of the courses. We have bioinformatics toolbox, thin biology for system biology. Like this, we have plenty of tools even for our applications. If you want to work with pharmacokinetics, we can able to work with the thing. Okay, now let me take you to the demo model, tumor growth model for file exchange. So this is the model I'm going to open using the thin biology tool present in MATLAB. So after opening this kind of a model, okay, where you can able to see in the form of a compartment model, this compartment model, you first build the after building this model, you can see the parameters here. Okay, you can see the parameters here. This is a property editor, and you can find central compartment, peripheral compartment, and tumor growth model. So rate constants are provided. We just simulated this kind of a model, whatever are simulated in the shown in the demo. So the properties parameters we can able to change it based on the properties and parameters. You can also see the variation with respect to the output. Now I simply go to the next step like model analyzer. After developing the model, then it will be getting like this. Paclitaxel, 30 milligram paclitaxel is taken every 4 days, starting on day 8. See, this is the thing we are using to check thing, and the stop time we have given is the 40 day. So up to 40 day, if paclitaxel is taken in this dose pattern, how about the treatment? Whether the R square value is close to 1 or not, that we can able to find it. In our case, we got R square is 0.99. It means that it is more precise and the paclitaxel is going to cure that kind of a patient. This is the final output. This is the final output with respect to the bioavailability of the paclitaxel. So I have completed even how to work with the pharmacokinetics using MATLAB. So I've completed my session. Any doubts you can ask me. Any doubts? Okay. If no doubts, tomorrow we'll be discussing on molecular talking. Okay. Have a great day to you all. Bye.